Hello knowledge seekers, welcome to another session of Fuzzy Scientist blog. So in this session, this is basically a quick video. So in this session, I'll be discussing about the reactions that are involved in the conversion of pyruvate to acetyl CoA, so which is catalyzed by pyruvate dehydrogenase complex. Which we have discussed yesterday. So I've also told that this enzyme basically requires 5 coenzymes, or simply put 5 cofactors for functioning of it in a proper way. So what are these 5 coenzymes or cofactors? So one is TPP, which is thymine pyrophosphate, lipoamide, NAD, FAD, and the last one is coenzyme A. So these are the 5 coenzymes which are involved or which do participate along with the pyruvate dehydrogenase enzyme in conversion of um, pyruvate to acetyl CoA. So let's look at it. Let's look at the reactions. What happens? So what basically happens is pyruvate reacts with TPP in the presence of enzyme pyruvate dehydrogenase. TPP is nothing but thymine pyrophosphate. So pyruvate reacts with thymine pyrophosphate in the presence of enzyme pyruvate dehydrogenase, resulting in the formation of carbon dioxide and hydroxyethyl thymine pyrophosphate. That means hydroxyethyl TPP. Simply hydroxyethyl TPP. So listen very carefully. So this hydroxyethyl TPP, which is formed in this conversion, now reacts with oxidized lipoamide. Oxidized lipoamide in the presence of enzyme dihydrolipoyl acetyl transferase, or simple sorry, you can call it as acetyl transferase or transacetylase because acetyl group is being transferred here. So the hydroxyethyl TPP now reacts with oxidized lipoamide in the presence of enzyme dihydrolipoyl transacetylase, resulting in the formation of TPP and acetyl lipoamide. This TPP, which is formed again, starts entering into the cycle of reaction with pyruvate, and the newly formed acetyl lipoamide enters into the next step. So in the next step, acetyl lipoamide reacts with coenzyme A and results in the formation of acetyl coenzyme A, which is acetyl CoA, our major resultant product of linking of this basic PDH, that means pyruvate dehydrogenase complex reaction. And also from this acetyl lipoamide, um, uh, one derivative is basically acetyl CoA and the other derivative is for lipoamide. So now this lipoamide undergoes an oxidation reaction in the presence of enzyme dihydrolipoyl dehydrogenase. This lipoamide, which is formed from the previous reaction where acetyl CoA is formed from acetyl lipoamide, is now undergoing oxidation reaction in the presence of enzyme dihydrolipoyl dehydrogenase, resulting in the formation of oxidized lipoamide. And you know what this oxidized lipoamide is used for. This oxidized lipoamide goes in reaction with hydroxyethyl TPP. So in the oxidation of lipoamide, the FAD plus basically undergoes is reduced to form FADH2. So and the important thing is the FADH2 formed here are used for the further reduction of NAD plus, which is another coenzyme involved in the PDH complex. So this FADH reduces NAD plus to produce NADH and H plus ions, and now these produced NADH and H plus ions are used or moved to electron transport chain, wherein they result in the formation of free ATP molecules. Thus, in the in the process of conversion of pyruvate to acetyl CoA, so we observe a result in the synthesis of 3 ATP. and as this reaction, as glycolysis ends with two pyruvate molecules, so we can interpret that six ATP are produced from this in linking reaction, or simply put, the reaction which is catalyzed by the PDH complex. So this is basically the reaction. So in tomorrow's video, I'll be explaining it more clearly with all the physiological process that happenings and the biochemistry, conversions or balances that happens during these reactions. Till then, see you.